Hello friends, welcome back to our CSIR-NET Life Sciences channel. In the previous video, we completed the detailed theory of molecules and their interactions. That included biomolecules, protein structure, enzymes, DNA, RNA, and different molecular forces. Now let us continue with a deeper conceptual explanation of molecules and their interactions, which is very important for CSIR-NET Life Sciences. First, let us talk about biomolecules in a broader sense. All living organisms are made up of four major classes of biomolecules: carbohydrates, proteins, lipids, and nucleic acids. These molecules do not work independently. Instead, they constantly interact with each other to maintain cellular structure and function. Carbohydrates mainly function as energy sources and structural components. Glucose is the central molecule in carbohydrate metabolism. It can be stored as glycogen in animals and starch in plants. Cellulose, on the other hand, provides rigidity to plant cell walls because of its beta-1,4 linkages. Proteins are the most versatile biomolecules. They function as enzymes, structural proteins, transport proteins, hormones, and antibodies. The function of a protein is directly related to its three-dimensional structure. Protein structure is organized into four levels: primary, secondary, tertiary, and quaternary. The primary structure refers to the amino acid sequence. Secondary structure includes alpha helices and beta sheets. Tertiary structure is the overall folding of a single polypeptide chain. Quaternary structure exists only in proteins with multiple subunits. Protein folding is mainly driven by hydrophobic interactions. Nonpolar amino acids tend to move towards the interior of the protein to avoid water. Polar and charged amino acids usually remain on the surface. Molecular chaperones assist in proper protein folding. They prevent aggregation and misfolding, especially under stress conditions. Misfolded proteins can lead to diseases such as Alzheimer's and Parkinson's disease. Now let us focus on enzymes. Enzymes are protein molecules that catalyze biochemical reactions. They are highly specific to their substrates. This specificity is explained by the lock and key model and the induced fit model. Enzymes work by lowering the activation energy of reactions. They stabilize the transition state rather than changing the overall free energy of the reaction. Enzyme activity is influenced by several factors including temperature, pH, substrate concentration, and inhibitors. Each enzyme has an optimum temperature and pH at which it works best. Competitive inhibitors increase the Km value without affecting Vmax. Non-competitive inhibitors decrease Vmax. Incompetitive inhibitors decrease both Km and Vmax. Now coming to nucleic acids. DNA and RNA are polymers of nucleotides. Each nucleotide consists of a nitrogenous base, a pentose sugar, and a phosphate group. DNA is double-stranded and usually exists in the B form. RNA is single-stranded and contains ribose sugar, which makes it less stable than DNA. Hydrogen bonds between complementary bases stabilize the DNA double helix. GC-rich DNA is more stable than AT-rich DNA because it has 3 hydrogen bonds. Molecular interactions such as hydrogen bonding, ionic interactions, hydrophobic interactions, and van der Waals forces are weak individually. But together, they provide great stability to biological macromolecules. These non-covalent interactions are reversible, which is essential for biological processes like DNA replication, transcription, and enzyme activity. Understanding these concepts is extremely important for solving CSIR-NET MCQs. Most questions test conceptual clarity rather than direct facts. Before moving ahead, remember that life at the molecular level depends on precise interactions between biomolecules. Even a small change in molecular structure can drastically affect biological function. That is why mutations in DNA can lead to defective proteins. Non-covalent interactions may be weak individually, but together they provide strength and flexibility to biological systems. This balance between stability and reversibility is essential for normal cellular processes. Enzymes are highly regulated molecules, ensuring that reactions occur only when needed. This regulation helps the cell save energy and maintain homeostasis. For CSIR-NET, always focus on understanding concepts rather than memorizing facts. Conceptual clarity will help you solve even the toughest MCQs confidently. In the next section, we will apply these concepts to solve more advanced MCQs. Please pause the video, revise the concepts, and then continue. Thank you for watching, and let us move to the next set of questions.